Hello, everybody. We're live for a very impromptu stream here. I can't believe I'm doing this. It's like 9:30 in the morning. Why am I doing this? Well, I'm doing this. Okay, good. I can hear myself. Great. Okay, so why am I doing this? Why in the world? I don't even think anyone's watching this. If you watch this later, hi, thanks for watching. So it was just brought to my attention like a few minutes ago that we got a new piece up on CMI from our friend Dr. Rob Carter on COVID-19 and viral evolution. Now, it may seem like I've been picking on Dr. Rob Carter a bit on this channel lately, which is kind of true, because he's like really prolific in terms of just putting stuff out at a fairly good clip. And it's so bad. It's so bad. And this is like right in my wheelhouse. And I just did a thing a couple weeks ago on the evolution of SARS coronavirus 2. So this was brought to my attention just a few minutes ago, and I was ranting about it to a few people on Twitter because it's so bad. So I'm just going to take a few minutes and go through this here for everybody, and I'll throw this up as a after I end it, I'll kind of put it officially as an installment in the Bad Creationist Genetics series. But let's just let's just go through this and see how bad it is. So first, is COVID-19 evolving? COVID-19 is the disease, not the virus. So that's the first thing. COVID-19 is not evolving because COVID-19 is a disease. The virus is SARS coronavirus 2. That's the one that's evolving. Okay, so that's first. Second, no, but it is changing rapidly. And we're going to go through this, and we're going to see how he talks about how there are mutations and blah, 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 blah. Well, do we have a word for when populations of biological entities change in their inherited characteristics over generations? Yeah, we have a word for that. It's called evolution, right? So is SARS coronavirus 2 evolving? Yeah, SARS coronavirus 2 is evolving, right? That's like, you could just stop right there. And there's a rule that I like, a rule of if you write an article and you want to make the headline a question, and the answer to that question in the headline is no, then you shouldn't write the article. So if the answer is no here, then just is X happening? Then just leave it. You don't have to do it. But Rob Carter saw fit to do it. So we're going to go through it. And this is short. This won't be a long stream. And again, I literally saw this, 15 minutes ago maybe, and I was ranting about it on Twitter with some friends. So now I'm going to rant about it to the void, because why not, right? This is kind of what I do. So this is completely unfiltered. I'm going to run through it and just real time my thoughts on this terrible write-up. So people are asking, is the virus evolving? Yes, it is. We've already talked about this, but he goes into, he says, explain the differences between mutation and evolution. Look, if the mutations are inherited, that's evolution. That's the definition of evolution, right? It's changes in frequencies of alleles over generations, right? That is the literal textbook definition of evolution, right? So it wasn't true for swine flu. I mean, yeah, influenza, what just happened? Oh, did I hit there we go. I just messed up my own screen, not your screen. Hang on, there we go. I clicked a link in the paper accidentally. Okay, it was true for swine flu. That's the 2001 H1N1 was swine flu. It is true for influenza, right? Although that may link to the 1918 stuff. Actually, I'm gonna open that link and just see what that links to, because I think it's the 19 swine flu. Nope, that's the swine flu from 2009 they're talking about. They're saying, no, that's not evolution either. It was. Peppered moths, Darwin's finches, these are all things that evolve, right? Allele frequencies change over generations. So how much change do we expect to see? I love right here that he starts with invoking edge of evolution. So I just did a little while ago a long interview with Dr. Michael Behe on this exact topic and talked about how Behe uses Plasmodium falciparum as an example of something that indicates a speed limit in evolution while ignoring the selection dynamics that it's experiencing, because Plasmodium adapting to anti-malarial drugs is what's called directional selection. You're imposing a strong selective pressure that overrides everything else. So how many mutations do we expect to see? Well, we expect it to adapt to be really, really good at dealing with those drugs, but that's going to swamp out any other adaptive evolution that might happen, right? Because that is the overriding selective pressure operating on that parasite. So that's a terrible example to be representative of some like global, rate limit for molecular evolution. What you need to look at, if you want to see just how fast can things evolve, is you look at events called adaptive radiations. When you have lots of available niche space and you have a population that spreads to utilize all those different niches, right? That's the kind of thing that's going to show you just how fast adaptive evolution can happen, right? So Michael B., he uses a bunch of numbers here. I'm not going to go through all the problems with his work. I covered it in the interview with him. He assumes constant fitness landscapes. He gets a bunch of like basic concepts in evolution wrong, including the definition of fitness. So suffice it to say, forget those numbers are useless. It's not worth considering. Let's move on. So we get down here, we just get like the litany of wrongs. So like there's always a waiting time problem. I don't know what that's referencing. I bet it's something Carter has written on the waiting time problem and that I and others have talked about. When they talk about the waiting time problem, they forget about things like recombination. They also ignore that evolution is a parallel process, not a serial process, right? So you can have lots of things evolving at once rather than single mutations in a series. Once you get rid of that, the waiting time problem goes away, right? Evolution change, multi-mutations, reasonable amount of time, waiting time problem, covered that. For things like viruses, high population size, if a specific change is possible, it should be expected. Right, because you have a population that is huge, you have small genomes and high mutation rates. So within one generation, you sample every mutation possible, right? He's right about that. You get basically every mutation possible. Fun aside, one of my favorite papers of all time featured a virus population in that exact situation. It's called Evolution Under High Imposed Mutation Rate. Adaptation Obscures the Load. It's by J.J. Bowl is the senior author, but he's not the first author. It's Springman et al., 2010. And that paper disproves genetic entropy, 100%, because you have this population of viruses that are being mutagenized, and they experience all the mutations. If Sanford is right, and that virtually all mutations are harmful, that's almost a direct quote from Genetic Entropy and the Mystery of the Genome. If Sanford is right, then those viruses should have gone extinct in that Springman 2010 study. Instead, what we found is something called a quasi-species, where you're constantly generating very high fitness genotypes. So you actually increase the maximum fitness of individuals within the population, but you're decreasing the average because everyone's living under high mutational load. So you have both selection, preserving those high fitness genotypes, plus you have mutations constantly finding them, but you also have lots of mutations constantly moving away from them. So it's a highly dynamic process, but the population reaches a dynamic equilibrium, not extinction, as was expected by the people who did the experiment. Again, that's Springman et al., 2010. Evolution under high imposed mutation rate, adaptation obscures the load, and it 100% disproves genetic entropy. So like you could just end the argument right there. Genetic entropy isn't real. That paper proves it. So let's continue onwards here. Is mutation the same thing as evolution? The answer to this question is no. I mean, mutation is one of the mechanisms of evolution. For anybody that's ever taken a basic biology class, you've got 5 mechanisms that violate what's called Hardy-Weinberg equilibrium, which is when a population is not evolving. And cool thing with these 5 mechanisms, you can remember them with your fingers, right? So you've got selection, right? Because thumbs up, right? Selection, yay, we're adapting, right? Index finger, gene flow, because you're going over there. It's migration, right? You're going over there, okay? We'll come back to the middle finger. Ring finger, non-random mating, right? That makes sense because the ring finger, right? Non-random mating. That's going to change your allele frequencies. Inbreeding is an example of that. That messes with Hardy-Weinberg equilibrium. Pinky, small finger, genetic drift, small populations, random changes in allele frequency, stronger in small populations. Pinky. That brings us to M for mutations, or if you want to do it the other way, you can do it that way too, right? Because they can be bad. So that is one of the canonical 5, like in your freshman biology class, mechanisms of evolution. By definition, mutations change allele frequencies in a population. Mutations are evolution. Like that's the end. It's just what it is, right? Okay. So that's not correct, Dr. Carter. Yeah, DNA, it's a molecule. It has biochemical changes, blah, blah, blah. We have repair mechanisms. Fine. Mutations are mistakes. Yes, we try to fix them. Okay. We're invoking the curse. Oh, like, come on, give me a break. Like, you don't need to do that. Okay. So now this is interesting. We get to some mutations are much more likely than others. Yes, we do. When mutations accumulate, we see a directionality. Yes, we do. The change over time is predictable. Sometimes, yeah. Human H1N1, let's talk about this because, oh, I love it. So this is looking at the change in base frequency in H1N1 over the course from like 1918 to whenever, right? This is from that terrible H1N1 genetic entropy paper by Carter and Sanford. And I love this because what's going on here is they claim that these changes are evidence of genetic entropy. And they're wrong because the changes that they're showing here are adaptive. And what are they showing? Look, you've got A in green, that's increasing, and U for uracil, because it's influenza is an RNA virus, so it's U rather than T, increasing, right? And then down here, you've got C and G decreasing. Okay, great. That's genetic entropy, right? No, that's adaptive. That's selection doing that. Why? Because in mammals, I think it's all mammals, we have part of our immune system. One of the things we recognize it's called CpG dinucleotides, right? A C followed by a G in your genome, and the P stands for the phosphate group and the phosphodiester bond. So it's CpG dinucleotides. We have things that recognize that, and when they see it, they go, that's not us, and it causes your immune system to flip out. Now, if you're a virus, do you want to have a lot of CpG? Do you want your host's immune system to flip out? No. What you want to do is minimize the amount of CpG so you can go into like stealth mode, right? That's what you ought to be doing. So what they're showing here is H1N1 adapting to being in a human versus a bird, right? Where you don't have this selective pressure. They're showing it adapting to its environment in humans by minimizing, by being selected to have less C and G. So you're going to have fewer CpG dinucleotides. This is adaptive evolution. This is not genetic entropy. This is not degradation. These are adaptive changes. And they completely ignore that in that H1N1 paper. And now it's 2021, and we're still completely ignoring that. Okay, let's keep going. So let's see. Yeah, this is due to chemistry. We know that. This is actually really interesting. I feel like someone's been reading my thesis because this is what my thesis was on. This is what I did in grad school. My thesis was all about the instability of cytosine and how that actually drives the evolution of a different group of viruses, single-stranded DNA viruses. They have there's some weird stuff going on. It has to do with cytosine being unstable, especially when it's unpaired. So you get cytosine on its own. It tends to do this thing called deamination, this loss of an amine, right? That's deamination, and it converts cytosine into uracil. And this is also one of the most important things in the broad evolutionary history of life, because this is one of the dead to rights pieces of evidence for universal common ancestry of the genetic code, or I should say of nucleic acids as the hereditary molecule, because this is dumb. Like, this is bad. If you could have like a way of not having to deal with cytosine, you would get rid of it, right? Because cytosine deaminates into uracil. Well, if you have your molecule, it's cytosine and it's uracil, I'm like, how do I know if it's uracil? How do I know if it's supposed to be cytosine? Well, the answer to that, it's so cool. It's so cool. It has to do with the origins of like these earliest mechanisms. Like, why do we use thymine? Because cytosine doesn't turn into thymine, it turns into uracil. So you can, I know I'm getting off into left field here, but you can use thymine instead. And then if you see uracil in your DNA, you just flip it back to cytosine. We have tons of enzymes. That's all they go around. That's all they do. They just take uracil in our DNA, remove it, and then put cytosine in there. Because anytime we see uracil, we know it used to be a cytosine, and it just did this deamination step. The fact that everything has to deal with that, and it's uracil and RNA, cytosine and DNA, that's like universal common ancestry. That's why that is. This also has implications for the evolutionary history of methylation, which I won't get into, but it's super cool. Let's continue. Mutations happen, species change. Yes, fine. Change is necessary for evolution. It's not enough. Change, no, see, they're conflating evolution with universal common ancestry, right? What he's saying is it's not evolution unless it's universal common ancestry. That's just like not what the words mean. Okay, that's just Dr. Carter misrepresenting, and I'm comfortable at this point saying misrepresenting rather than getting it wrong. Like, he knows better, right? We can all agree on that, I think. He knows what evolution means. He knows it's not the same thing as universal common ancestry. Okay, like, let's be clear on that. So there's no mechanism to produce blah, 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 blah. This is why we're comfortable talking about change. Mutation is not the same thing as evolution. He's just like repeating the mantra, right? Again, and this is something I've talked about before, I think, that this is not aimed at me. This is not aimed at, convincing scientists, convincing evolutionary biologists or geneticists. This is repeating a mantra, right? This is open your hymnals to, whatever for today. We're going to talk about how natural selection is not evolution and mutation is not evolution, right? This is for the people already on board to reassure them and to give them talking points, right? That's what this is an exercise in. It's not an exercise in conveying scientific information, and obviously not for a scientific audience, right? So that's what we're doing here. And you see that in the structure, right? It's very repetitive, right? Okay, so natural selection is not the solution. So viruses are at war, blah, blah, blah. Only the strongest survive, not as robust. So this is where we get into some really interesting stuff, because this is similar to the error that Carter and Sanford make talking about H1N1, where they say that a decline in virulence is evidence of genetic entropy. What they're ignoring is that in the ecological context of a pandemic, this is actually adaptive. And it's not that the viruses just get worse. The word they use, we'll see if they use it here, but they use it for H1N1, is attenuated. That, it attenuates over decades and it gets worse at making humans sick. No, that trait of making people sick, that's called virulence. And that's something that you could select for, right? That's a phenotype, right? It's all about the interaction between the virus and the host. And that's a phenotype that you can select for. And it's going to be, under some circumstances, under selection, to be more virulent, to be more dangerous to the host. Under other circumstances, it's going to be less virulent. It's going to be under selection to make the host less sick. And as you progress in a pandemic, as the frequency of susceptible hosts decreases as a percentage in the population, you're going to be under selection to persist in your host for a longer period of time to facilitate greater transmission, to make it more likely that you're able to spread. As a side effect to that selective pressure, you often see, not always, but you often see that viruses become less virulent. I just did a stream on this a couple weeks ago called Evolution Explains the Delta Variant. I go into this dynamic in a lot more detail. It's a little bit long. It's like 40 minutes. But if you want more on that kind of dynamic, check out that stream. It's got lots of details on that whole evolutionary situation. So let's bring this back to SARS coronavirus 2. We see a similar pattern, tabulate all the mutations, CT mutations dominate. Fine, I'll take him at his word for that, because because as it's adapting to humans, what's it going to do? Minimize CpG dinucleotides. We already talked about that. Those changes are going to be adaptive for the virus. So we see a bunch of changes. The new strains are not proof of evolution. Fine, okay, I guess. Basically, what we're seeing is that the Delta variant is more transmissible. It might be more dangerous. I haven't seen these data specifically, but it's like approximately as dangerous as the original strains if you're not vaccinated, maybe more, maybe less. It's definitely less dangerous if you are vaccinated. Get your vaccines, people. We just got full FDA approval for Pfizer yesterday, and Moderna's coming down the pike soon. So, get your vaccinations. Let's end this pandemic. Let me just throw that out there. So what we're seeing is enhanced infectivity. Oh, that sounds like beneficial, right? That sounds like it's beneficial, right? That sounds like instead of genetic entropying and getting worse, it actually sounds like it's adaptive and it's getting better. That's interesting. Now, So let's see what we see here. Vaccines are still protective. Yes, it is. I don't know if this is true. I'm skeptical that we could actually have a true escape kind of strain that the vaccines completely miss, because you're messing with that spike protein. We don't need to get into the biochemistry. It doesn't matter. I don't think it's likely, because you would change it so much it wouldn't match your cells as well. We don't need to say more than that. I'm skeptical of this claim, but that's kind of to the side. We don't care about what specific mutations. We don't care. What I want to focus on, we get right to this end paragraph, and this is where we get to it, because this is where Carter messes up. This is where Dr. Carter messes up, I think. So here we go. Changes like this are expected. They do not mean the virus. I mean, it literally is the virus evolving, first of all, right? Because it's changes in the genetic makeup of the virus over generations, right? It's literally what evolution is. In fact, over time, viral infectivity should wane. I mean, by infectivity, do you mean virulence? He's not defining his terms well, but infectivity usually means transmission. And what we tend to see is selection for greater transmission as you vaccinate the population, because susceptible hosts are harder to come by unless you live in Florida. Florida, get your stuff together. Get vaccinated, y'all. Now, each viral strain picks up more and more mutations. It should become less and less robust. This is where they're wrong. So that's not true, because what we saw in H1N1 is that it was adapting to the new ecological conditions of that pandemic. As H1N1 circulated, remember, this was starting in 1918 and going further on. What we see in that is it's adapting to a situation where there are fewer and fewer potential hosts around. So you have a selection for inter-host competition at the expense of intra-host competitiveness. Now, that's what we're seeing in SARS coronavirus 2. And again, I talked about this in Evolution Explains, the Delta variant. So this took decades in H1N1. SARS coronavirus 2 only been around for a short time. Will genetic entropy degrade the virus? Yes. Citation needed. Now, there we go. Citation needed for that one, because like genetic entropy isn't real. Will it happen this year? No. And while it is degrading, it might pick up mutations that make it spread faster, or one that makes it more deadly. Now, here we are, right? This is really interesting to me, because what he's saying right here is that as this virus is degrading, as it's genetic entropying, it might get better. It might increase its fitness. And this is in stark contrast to what Dr. Sanford claims in Genetic Entropy and the Mystery of the Genome, because he's very clear about this. He's very clear that this is, and depending on the chapter in the book, he describes it as a linear decline. In other places, he shows it as an exponential decline, but it's a decline. It's not a, up and down, and like maybe over time the trend is down. It is just down. And what Dr. Carter is saying here is, well, we start up here. Eventually, we're going to get down here. But in the meantime, like, it could go up, it could go all over the place, right? It might even get better for a long time. That's violating genetic entropy in a couple of ways. One, it means that beneficial mutations are real and selectable, and are a large enough fraction that they're actually going to happen. Sanford says you can basically discount them. Carter is saying, no, beneficial mutations are actually a thing that can happen, and we'll detect them, and they can affect how this thing actually evolves. Two, if you're going to go from a situation where you have beneficial mutations to collapse, something has to change, right? So you have to have a situation where other mutations cause the effects, swamp out those beneficial mutations. You have to have something worse, or maybe he doesn't say, but maybe there's antagonistic pleiotropy in those beneficial mutations, and they also have some downside that will become apparent later on. Or maybe the ecological situation changes, and those beneficial mutations are beneficial now, but then later on they'll become a problem, right? And the population will go extinct. The problem is all that stuff, that's not part of genetic entropy. Genetic entropy is very straightforward. Most mutations are harmful. As mutations accumulate, population gets worse, eventually it goes extinct. Very straightforward. And when I say most mutations are harmful, I mean virtually all. Right? The fundamental thing that's going on here, other than not defining evolution correctly, is that Sanford, and I think what Carter is building on here is the same thing, is that they define fitness as biochemical complexity or biochemical functionality, rather than reproductive success, right? They're just basing this whole idea on the wrong definition of another fundamental term in evolutionary biology, fitness, right? So they're saying it's all about complexity or information, which they can't define, rather than reproductive success. But at the end of the day, what we're seeing is evolution in SARS coronavirus 2, just like we saw in H1N1, that was adaptive in the current ecological context, which means early in the respective pandemics, you have selection to get into your host and kind of be a bull in the china shop. Later on, either decades later, years later for H1N1, it didn't take decades for the 1918 strain to become not super deadly. It only took a couple of years. But over that time, what happened is lots of people were infected. You decrease the host frequency, right? Because people had been infected and weren't potential hosts anymore. So you had a flip in the selective pressures. Same thing is happening here, and it's being pushed along faster by mass vaccination, right? Which is another reason we need to vaccinate everybody, because this is pushing this virus to a place where it's potentially not going to be as dangerous, right? So that's kind of the end. I don't have a nice, neat wrap up for this, because this was just a completely impromptu, like, Carter said something silly and I'm mad about it on Twitter, so let me just throw up a stream and go through it. That's why this paper is bad. It misdefines basic terms, gets basic facts about like the H1N1 evolution wrong, ignores how certain aspects of their molecular evolution is actually adaptive in the context of infecting humans, and ignores how adaptive evolution actually occurs during a pandemic and how the selective pressures change early versus late in a pandemic. Rob Carter's just saying all that stuff goes under the heading of genetic entropy. Eventually, we swear. And also admits that there are beneficial mutations happening here, which is something that Rob Carter does not accommodate in his, I should say, selectable beneficial mutations happening here, which is something Dr. John Sanford doesn't accommodate in his version of genetic entropy as presented in the book, Genetic Entropy and the Mystery of the Genome. So that has been me ranting about, is COVID-19 evolving? No, but it is changing rapidly, by Dr. Rob Carter on CMI. Thank you for those of you who brought this to my attention. You know who you are. I appreciate you. Hope everybody enjoyed that Tuesday morning rant on a really bad bit of creationist genetics. Hope everyone has a great day. Bye, everybody. Now I just have to find the end broadcast. There it is. There's the button. All right. Bye, everybody.